Please welcome with me on stage Stefan Borgwardt.
Hello, everyone. Nice to see you. Great that you appeared even after lunch. So hope to entertain you a little bit. So you've seen the title of my talk. I'll speak about distinct acute effects of LSD, MDMA, and D-amphetamine on brain connectivity. So as Philip already told you, I won't speak about connecting people. I will speak about connectivity, so slightly different, but there are some similarities. So why do I speak to you? As a psychiatrist, I'm mainly interested in treating and helping people. Why? Because mental disorders, neuropsychiatric disorders are widely spread worldwide. They have a great impact on society, and we are able to help many patients, but not all. So there is a need, an urgent need, to develop safer and better treatments for our patients. So that's my motivation to get into this field. You see 3 maps, and maybe you can now start to look at these kind of imaging maps. Maps with it's not a gray matter map or white matter map. That are not brain maps, that are maps from towns. And I will present data from Basel on the left side. I've been trained in London. This is the River Thames, and now I'm based at the University of Lübeck, and you can see these 3 rivers. First of all, I'd like to especially thank a few colleagues of mine. Matthias Lichty, he is around us, I guess, at least he was around. We started more than 10 years ago, I think in 2010, when we thought about how to image acute effects of psychedelics in the brain. And especially I'd like to thank to Felix Müller and Mihai Avram, colleagues from Basel and from Lübeck. And they mainly did most of the work, I have to admit, at this stage already. So I don't need to show you these persons here, I guess. Sorry, I was a little bit distracted. These are the 3 colleagues I'd like to especially thank. And I don't need to mention these 3 persons who are famous for consuming psychedelics or trying to consume psychedelics. I will mainly speak about these 3 topics, and I try to address at least the questions. Also, I'm not able to answer all these questions fully, but I will address them. So how does MDMA work in the brain? How does LSD work in the brain? Is LSD in some way special to other substances? I will shortly present some data on LSD for depression, and will give an outline about future studies and planned studies. So what was the first imaging study on LSD? This study, really a pioneering and landmark study, was published in 2016 by Carl Harris and David Nutt's group in PNAS, and this was really, to my knowledge, the first neuroimaging study in modern times of LSD. And they investigated 20 healthy subjects, provided LSD or placebo, and measured different imaging modalities like cerebral blood flow, resting state activity, and MEG. And what they found was an increased visual cortex cerebral blood flow and an expanded visual cortex functional connectivity profile. And most interestingly, they did not only had this fancy blobs or blobs marked here in red, They also showed a correlation towards ratings of visual hallucinations. So the increase in the functional connectivity was related to symptoms. And this is always very important if you look at imaging findings, that you don't only look at the blobs, you always want to see some sort of correlation with symptoms, with clinical outcomes, whatever. But otherwise, at least from a clinical perspective, is that it doesn't really make sense to do imaging. So that is one requirement you always want to see. So this was the first imaging study, and so as I said, our studies in Basel have frequently, but not exclusively, focused on LSD. We have studied the acute effects, mainly using functional magnetic resonance imaging. I will say a few words about this method now. And we were in particular interested in two target values, in task-evoked activity and in functional connectivity. So what is fMRI? I think you have seen in previous talks from Drummond and others here at the conference that functional MRI is a very useful tool in identifying areas of cortical activation and also in discerning areas of neuronal connectivity when used during resting state. But the basic principle behind fMRI relies on the dynamic response seen in cortical areas and the different magnetic properties of oxygenated and deoxygenated hemoglobin. And if you look at this picture here, whenever a task is applied, so if you do something, if you listen to a speaker somewhere, then you have to concentrate and you have to do some cognitive work. And during this work, there will be an increased cerebral blood flow and a hemodynamic response, and the properties of the hemoglobin will change. And you can track this change and map this into signals, and at the end you get fancy maps and blobs where you can give an indirect measure of the neuronal activity. And so a relatively simple example of a task-evoked activity is to provide the task open eye or close your eyes, and when you do that and scan someone, then you see an activation in the responding or responsible brain area here, the visual occipital cortex. And we did that in healthy volunteers. So we thought to apply a task which measures emotional processing, or which reflects emotional processing. And emotional regulation plays a crucial role in the clinical effects of psychedelics. So neuroactivation to fearful phases, so we presented fearful phases to the patients in the scanner against neutral phases after placebo and LSD treatment was measured. And as you see here in a very small plot, LSD reduced the neural response to fearful phases in the amygdala, you can see here, and the right medial frontal cortex, both regions which are implicated in emotional processing. And interestingly and importantly, these LSD-induced changes showed a negative correlation between the amygdala activation on one hand during the presentation of fearful phases and the visual analog scale measurements of subjective drug effects. So again, there is a correlation between the imaging findings, the amygdala activation, and the subjective drug effects, the clinical measures. We also applied another task, a response inhibition task. We thought to apply this because we know from different substances used as drugs, that they have some sort of effects on response inhibition. So we measured this, and we found again a relationship between the activation in a region associated with response inhibition, here the parahippocampus gyrus, and the probability of inhibition between placebo and LSD. And again, there was a negative relationship, negative correlation between the severity of these cognitive deficits, you could say, or cognitive impairments, the reduced response inhibition, and the activation in the right medial frontal gyrus and the superior right frontal gyrus. These findings show that the serotonergic related activation by LSD leads to a hippocampal prefrontal cortex mediated breakdown, if you want, or inhibition of inhibitory processing, which might subsequently promote the formation of LSD-induced visual imageries. So that's a conclusion out of these findings. So going away from these task-related fMRI findings, we looked at the connectivity. And connectivity actually means, or functional connectivity means, that there are correlations between signal trajectories over time of different brain regions. So when you find a kind of temporal association between different brain regions, that's called functional connectivity. And in the brain, there are different networks. You may have heard of some of them, for instance, the default mode network and others, sensomotoric network, visual network. These brain regions work functionally together. And we were interested in the effects of psychedelics on these measures. And here I show you a slide when we applied MDMA, so not LSD, MDMA. Here I'd like to start with MDMA first. So we looked at 10 available brain networks, intrinsic brain networks, and looked what happened when participants got MDMA instead of placebo. And we found that we found a significantly decreased connectivity within some of these networks, here the default mode network and visual networks, and sensory motoric networks. And in contrast, we found increased connectivity in a different network. And we were kind of surprised of this result, because this result was more or less identical to the one which we have seen in LSD. So the non-hallucinogenic drug or substance of MDMA had similar results with respect to functional connectivity compared to LSD. And if I show you the next slide, then you can follow this. So again, when we applied LSD, compared them to placebo, we found this decreased connectivity in the default mode network, visual networks, and sensory-motoric networks. This was interesting, that they were on one hand identical or similar to the results of previous studies by Harris, which reassured us that these findings might be real. But on the other hand, we found it quite interesting that this was similar to the one in MDMA. We now looked at connectivity that was not connectivity within these networks, we now looked at connectivity between different networks. You could say we were now interested in a broader sense. So what do these substances to different networks in the brain? And here we found significantly increased connectivity between numerous, or you could say almost all networks in the brain. So LSD seemed to increase these between network connectivity over the whole brain, in contrast to MDMA, which did not these increase in the between connectivity. So LSD seemed to decrease the segregation of the different various brain networks, and by increasing the connectivity between these networks, as explained. So in the next step, we were not just we were interested in a more global functional connectivity perspective. So we now looked at the average correlation of each voxel in the brain with other voxels of the brain. So we had no a priori hypothesis what region is in particular interesting. We now looked for the global functional connectivity. And as others, we found that following LSD administration, there was an increased connectivity in the area of the thalamus and certain other brain regions. And again, these results were consistent with other studies that used this method. And you can have a look at these more or less fancy figures that illustrates the functional connectivity. Oh, sorry. No. The functional connectivity between the thalamus and different regions in the brain. And when participants got placebo instead of LSD instead of placebo, then the functional connectivity was increased almost over the whole brain. So this study and others suggested a hyperconnectivity between the thalamus as a kind of gatekeeper of the brain and different areas in the brain, as here sensory-motor regions. And again, this connectivity measure was associated, significantly correlated with some sort of clinical measures here, visual and auditory perception. As I told you before, so my background is a little bit in the field of early detection of psychosis. So I was very much interested in my first years and up to now how psychotic disorders develop. So I found it interesting to see these hyperconnectivity between the thalamus and certain brain regions here in our LSD study, because I thought, okay, we have heard this story in a way in psychotic states. So my colleague Miha Avram, he discussed the thalamic filter theory, which is established in different fields in cognitive neuroscience, for psychotic states and for psychedelic states. And he found that the hyperconnectivity might be a possible candidate for a common neuronal psychotic and psychedelic condition, which is interesting from my point of view. We were, in the next step, interested to directly compare the effects of LSD, D-amphetamine, and MDMA on the thalamocortical connectivity. And we found that this connectivity was the connectivity between the thalamus and the sensorimotor cortex does not only occur with LSD, but also with the other substances. You can see here. So almost same activation or hyperactivation across these substances. The LSD changes were correlated with various measures, various clinical measures. Here, blissful state, insightfulness, changed meaning of perception, all these clinical impressions and sensations people who take these substances feel. But interestingly, we found no significant correlations between the connectivity and any kind of clinical symptoms after administration of amphetamine and MDMA. So we wondered ourselves, what drives this thalamocortical hypo and hyperconnectivity? Does various cortex types may reflect different neuropharmacological effects or not? So, and my colleague, he did lots of fancy analysis, but basically he found that there was, and this has been discussed in the talks yesterday and yesterday before, that there seemed to be an increased bottom-up and decreased top-down information flow between the thalamus and some sort of cortices across all three substances. So this was a kind of common pathway or similar effects of these substances. But if you look not on specific unipolar cortices, if you look at more or less the whole brain again, transmodal cortices, then you found an increased effective connectivity of the thalamus to all cortices, which indicate that only LSD breach the hierarchical organization of the ongoing brain activity. So to make this complicated story a bit simpler, I think you can interpret that LSD interferes much more into the organization and connectivity of the whole brain compared to MDMA and amphetamine. You have seen this slide before, but again, I want to show you this because this slide published by Vollenweider and Preller 2 or 3 years ago in Nature Review Neuroscience, that summarizes this kind of model, how we understand the psychedelic-induced brain activity. And as you see here, the thalamus and the thalamic filtering plays probably a crucial role in the effects of psychedelics. And this is shown here by the, or through the reduction of the thalamic filtering, there is an increased connectivity between sensory cortices, so the cortex on one hand, and the rest of the brain. And this leads to some sort of increased sensory processing and the psychedelic experiences. This was functional imaging. And in the next step, I want to show you some ideas or some thoughts about structural imaging. We haven't heard, I think, here during this conference about structural changes. And this is kind of understandable because there's a lot or less research on structural changes after acute effects of psychedelics, of course. But we thought that it's worthwhile to look at the brain structure as well. And we used here diffusion tensor imaging. Some of you may know this. It's a measure of the water diffusion in the brain. And the brain you can divide into gray matter and white matter. And the white matter can be tracked by diffusion tensor imaging. I won't go into detail here. But there are two measures of white matter structure. One is the FA and the MD. And to make a long story short, you can say that the FA, the fractional anisotropy, summarizes microstructural changes, whereas the MD reflects the mobility of water molecules and is or could be a correlate of axonal degeneration. And you have probably heard in the talk of Jasmin Schmid yesterday about the LSD study in depression. So now we switch from findings in healthy volunteers to findings in clinical population. And this study was done in Basel. It was a crossover design. And the idea is to provide two dosages, low dose or high dose of LSD, 4 weeks between the administrations, and 61 patients were treated, and 35 of them had a DTI scan. I won't go into the details of the clinical findings. I'm sure you have seen this yesterday. So in short, LSD reduced the severity of depressive symptoms over time significantly, which is of course a very important finding. But we were mainly interested how is this reflected by structural imaging. And as I said, we looked at FA and MD in these patients. And we found a modulation of the FA, of the white matter structure in some brain regions. And it's interesting to see, and I don't know if you had expected these findings after relatively short periods of time. So, you know, the follow-up, the follow-up time were up to 19 weeks. It's not too bad, of course, for a clinical study. But I don't know if you had expected that there are structural changes in the brain after this relatively short time. But in this study, there were associations between the fractional anisotropy and clinical variables. And so we measured the difference between the depressive symptoms over time, 9 weeks to 2 weeks, week 2 to 9, week 2 to 13, week 2 to week 19. And you see that there is a correlation between the FA and the decline of depressive symptoms over time. And this effect got even stronger over time. So there was a significantly, statistically significant correlation already at week 9, but it became even stronger, or was very strong, at the end of the follow-up period. So you could say that 2 moderate to high dosages of LSD increased the FA for patients with major depression. And again, important, the FA modification correlated with the improvement of symptoms. And this study indicates or shows, as you like, that LSD is able to modify neuroplasticity in the brain, and you can measure it, and even after a relatively short time. So what are the next step? What are our planned projects? We think we will focus our study a bit more on a very important clinical outcome as suicidality. So here I showed you effects on depressive symptoms, which is always a mixture of different symptoms. But one of the most obviously important clinical features is suicidality. So we want to look at these maybe specific effects or beneficial effects of ketamine and psychedelics. We want to implement more translational clinical offers in our department. Compassionate use is possible in Switzerland. It's not possible in Germany, unfortunately. But I'm sure that since ketamine has now been really and widely established in clinical utility now, so we treat many patients each day with ketamine, it won't take long time that the gap between the provision of healthcare services in Germany compared to other countries will get a little bit smaller. And I was very much inspired by the talk from Tessin yesterday, where he spoke about the real-world data acquisition. So I think that is another very important step forward to gather, apart from RCT research and findings, also to gather real-world data. And another topic we are very much interested in is to compare the antidepressant effects across several psychedelics. You have seen different results from studies or from colleagues who were interested in MDMA or in psilocybin or DMT or LSD, but we haven't seen so much research on head-to-head comparisons between different substances. And we were interested in some more mechanistic approaches. So there is a lot coming up, hopefully. But before the future comes, I'd like to thank my colleagues in Lübeck, and I already mentioned my previous colleagues in Basel. So this is my small translation psychiatry group, and I'd like to thank you for your patience and interest, and of course all our patients and participants and the funders. And I'm happy to take questions from you. Thank you.
Thank you very much, Stefan. The 2 microphones are ready, and we have time for around 2 to max 3 questions.
Hello. So first thing, thanks for your talk. I'm here.
Yeah, thank you.
So I was thinking, do you think it will be worthwhile, and if we could draw a correlation between brain connectivity and neuroplasticity and HPPD, reported HPPD effects?
Can you repeat?
Okay. Do you think we could draw a correlation between brain connectivity and neuroplasticity and HPPD reported effects? HPPD.
Can you explain?
People that take psychedelics sometimes have flashbacks and HPPD after. And
Yes. We published a paper on the importance to look at this flashback phenomena. Yes, that's very important. So, but what you probably need for that is larger sample sizes. So the studies I've shown you contained 20, 30, up to 60 patients as participants, and as these phenomena are not that frequent, you would need probably larger sample sizes, but it's very important, yes.
Okay. And so the question was, do you still observe higher connectivity years later after intake, for example, a year or two?
I don't know, because we have no data yet.
Okay, thank you.
Hello. Over the last couple, I'm over here. Over the last couple of days, it seems like a lot of the research has gravitated around the same established compounds, LSD, MDMA. They've been around for decades and decades. But there's also quite a few interesting emerging compounds too. Are there any plans on the radar to explore these a little bit more, especially in terms of connectivity?
I have no plans, because I have a lot to work to get these established compounds to Germany. So that's difficult enough. But I could imagine that whoever has a possibility to investigate newer compounds and new and upcoming compounds may use connectivity as a kind of marker, biomarker, and use these methods. I could definitely imagine. And what you've seen is that between connectivity, there is a much better correlation towards clinical symptoms, as you have in other imaging methods. So I've done loads of structural imaging studies, looking at gray matter volume, gray matter, whatever, and we sometimes or often struggled to find a correlation. So we have seen people with gray matter disturbances very marked, but almost no clinical symptoms, and the other way around. And here with connectivity measures, it seems that you have a better indicator or biomarker which is more related to clinical symptoms.
Hi, thanks for the talk. Just related to your connectivity findings, for your DTI functional angiotropy, did you use the DTI data to construct the structural connectome of the brain and then see if there were any changes in the structural connectome per se, instead of just the voxel-wise changes in FA?
No, not yet. No, but I will take this inspiration with me. Yeah. Let's talk about it. Yeah. You showed these nicely Copenhagen database, and I think it would be worthwhile to use these data. I mean, we haven't published these. We have submitted this paper, so it's kind of new. So it would be very good to look at structural connectome. We have done these with psychosis patients, but not in this study yet. Very good idea. Thank you.
I'm really sorry. Thank you, Stefan. I was kindly asked to be very strict with the timing, since Stefan and his daughter have to catch their ride back home, basically. I'm really sorry, but we will have to cut the Q&A here. Thank you very much, Stefan.
Thank you.
And we will continue with...
Thank you very much. Thank you for having me here. It was an excellent conference. I really admired the interdisciplinarity and diversity of this audience and of the whole conference. That is really special, different to other conferences. Really like that. Thank you very much.
Thank you, Stefan. That's very nice.